Welcome to LankanTutor's Biology 2.8. If you're not watching this at LankanTutor.com, we suggest you do so, as it has a bunch of written tutorials that could possibly help you out. And we're here with one of my favorite parts is the cellular basis of life. The part we're going to study about today is related to organelles, and it's going to be rather long because we're going to be covering the entire structures of eukaryotic cells, prokaryotic cells, differences between plant cells and animal cells, and every single structure inside the cell and their purpose. So this is going to take a while. So let's begin. First of all, you need to realize that what a cell is. The cell is the smallest living unit. It is the basic structural and functional unit. We discussed that in the first part of biology in, I think it was 1.0 or it was 1.1. And anyway, before that, we need to realize something. If you remember last time, I talked about two people called Robert Hooke and Anton van Leeuwenhoek who used the microscope to find, well, single-celled organisms and cells. Basically, those were the people that first found the cells. You don't really need to know a lot about them. Just remember that Robert Hooke was the guy who first found the cell, and he's the one who named it as the cell. And Leeuwenhoek was the guy who found the first unicellular organism, a organism which is a single cell, and that organism happened to be a euglena. I'm going to write these names down because I'm not sure if I'm pronouncing them properly. Then I believe his name is Matthias Schilden. He found out that all the plants were made out of cells, and Theodor Schwann found out that animal tissues also comprised of cells. And eventually, this led to a theory called the cell theory. So Schilden and Theodor Schwann were the people to introduce the cell theory, and what this theory was that three basic principles about the cells. The first is that all organisms are composed of one or more cells. If it is less than a cell, it is not an organism. So remember, the cell theory, you need to memorize that. There's no way around it. All living organisms comprise of one or more cells. The second part is that the basic structural and functional unit of an organism is a cell. So if you ask what a cell is, this is actually the best way to define it. It's the basic structural and functional unit. And remember that it is also a part of the cell theory. And the final part of the cell theory is that all cells arise from pre-existing cells. So cells divide to form new cells. So since we got that out of the way, we're going to have to talk more detail about the cells. To start it off, we're going to divide these cells into two categories. Now, these categories are depending on the complexity of the cell. The first category, we're going to call it the prokaryotes, and the next category is going to be called the eukaryotes. The difference between, well, the main difference would be that the eukaryotes have an organized nucleus. If you don't know what a nucleus is, the nucleus is the place where all the DNA and matter gets stored. It's like the brain of the cell. And the prokaryotes have an unorganized nucleus, which means it's nowhere near as complex. It's found in circular DNA. They're not arranged properly into the manner that we could find it in eukaryotic cell, which is extremely complex, and it is a really complicated structure. So let's go into the prokaryote a bit. Now, we know that the prokaryotes are less complicated cells, and they are usually bacteria and archaebacteria. So since they're not really complicated cells, naturally the size of these cells would be smaller. So the size of a prokaryotic cell is generally around 0.2 micrometers to 0.5 micrometers. And they don't have any membrane-bound organelles. So they don't have any structures like chloroplast and mitochondria and a nucleolus, because those structures have membranes. And prokaryotic cells, the main feature, besides the fact that it doesn't have an organized nucleus, is the fact that they don't have membrane-bound organelles. So what do the prokaryotic cells have? So we're going to start drawing the model structure of a prokaryotic cell. This is what you need to be able to draw if they ask you what the prokaryotic cell is. Now, we know that it doesn't have a membrane-bound nucleus, and the material inside, the genetic material inside, is presented by coiled up circular DNA. You need to remember that it is a circular DNA, but even though it's not covered by a membrane, it is generally centered. It's in one specific area, and that area is called the nucleoid. Instead of calling it a nucleus, you call it a nucleoid because it does not have a membrane. The membrane in the eukaryotic cell is called the nuclear envelope. In addition to a nucleoid, small circular DNA are present in the cytoplasm, and those ones are called plasmids. So if it's not in that centered area, it's not in the nucleoid, they make separate structures called the plasmids. Prokaryotes don't have a cytoplasmic skeleton, and organelles like endoplasmic reticulum, also organelles like mitochondria and Golgi bodies and lysosomes. The ribosomes are present in prokaryotic cells, but the thing is, unlike the ribosomes present in the eukaryotic cell, they are much smaller. In fact, to be precise, the ribosomes present in a general eukaryotic cell would be called an 80S ribosome, whereas in the prokaryotic cell, it would be called a 70S ribosome. There's a rigid cell wall around the plasma membrane, and in bacteria, it's made out of this material called peptidoglycan, which is also known as murine. But in archaebacteria, polysaccharides and proteins are present in the cell wall. Usually the cell wall is enclosed again in a capsule. So remember that the plasma membrane is enclosed in a cell wall, which is enclosed again in a capsule. Some prokaryotic cells possess flagella. Now, flagella are these long strands which help them move. It's an appendage used for locomotion. Remember that not all of the prokaryotes have that. Some may have that, some may not have it. And there are also other minute thread-like structures called the pili, which also help it attach to surfaces. Just like flagella, it's not found in every organism, but it could be present. So that pretty much covers up everything you need to know about the prokaryotic cell. And we're going to go into the eukaryotic cell, specifically the plant cell and the animal cell. You need to realize that these two cells are completely different. Actually, every cell is slightly different to each other. What we're doing is we're giving you the basic model structure so you can identify if several of these features are present in a cell. You can classify that cell into, well, a plant cell or an animal cell. So we're going to go ahead and begin with the plant cell, and I'm going to start drawing it now. The first thing you're going to see is that it has a specific shape. That's because the plant cell has a cell wall. This is really important because animal cells don't have cell walls, and in the cell walls that the plant cells have, they could have primary cell walls, and sometimes they could have secondary cell walls. Now, everything I'm writing down, you need to memorize this stuff. It's really important. The primary cell wall is made out of a structure called cellulose. We discussed that in the elemental composition of how cellulose is made and all those stuff. You might want to check that out if you don't remember. So it's made out of cellulose and hemicellulose and pectin. Those are the three main constituents that could be found in the primary cell wall of the plant. Now, the reason that we call it primary cell wall is because some plants have a secondary cell wall, and the secondary cell wall is made out of substances like suberin and lignin. Another thing you need to remember though, that the plant cells are living cells. The cell wall of these cells are dead. They do not have life. They're basically just there for protection and to give the plant rigidity so it doesn't collapse on its weight. So I'm just going to throw in a bit of terminology in here just so you don't get confused of what I'm about to say next. The protoplasm is the living content of a cell that is surrounded by a plasma membrane. It's the general term for the cytoplasm. So the protoplasm is composed of a mixture of small molecules like ions, amino acids, monosaccharides, water, macromolecules, and nucleic acids, proteins, lipids, polysaccharides. In eukaryotic cells, the protoplasm surrounding the cell nucleus is known as the cytoplasm. So don't get a little confused when I say the words protoplasm and cytoplasm. It might be a bit confusing, but it's generally, for now, it's kind of like the same thing. So we discussed that the plants have a cell wall which gives it strength and so on. But if the plant cells are connected to each other with a lot of cell walls, how does anything work? Are they separated from each other, or are they in a network? Now we're going to go ahead and zoom in on the cell wall of the plant. I'm going to go ahead and label the parts. This is called the middle lamella, and then there are intracellular air spaces, and this is the primary cell wall. We're not drawing a structure with a secondary cell wall. Now, in the primary cell wall, there are places which are thinner than the normal places, and these are called pits. And you can see small pores in the pits which connect the two cytoplasms. And there are things called plasmodesmata. A plasmodesmata is the protoplasmic strand that connects two adjoining cells. So you might want to remember that, and using these plasmodesmatas, the cells build these intricate networks, and they send water and ions and all sorts of things through the cell. Now, the other thing is that the primary cell wall is a fairly thin and permeable cell wall. Now, that permeable means that it has the ability to send water and some solutes through. But the secondary cell wall contains lignin and suberin. Now, because of those layers, it is completely impermeable and nothing can go through it. Now, there are pros and cons of having a layer of suberin. The thing is that because of this, it cuts contact with everything else, and the layer generally dies off. The cells surrounded by lignin die off. But unlike cellulose, it is extremely hard, and because of that, it gives so much more strength than a primary cell wall, and unlike the primary cell wall, it's not that thick. But then, how would plants without a secondary cell wall stay upright? Because we discussed that not all plants have a secondary cell wall, and that these plants with secondary cell walls tend to kill off the cells which are covered by suberin and lignin. Now, when a column of water is pulled up through the plant's xylem, don't worry, we'll get into the detail of how the transportation works, but I'm just giving a brief description so that it would be easier when we're at the 6th lesson, where we'll actually discuss about how the transportation and translocation and everything works. So it's by this thing called turgor pressure, the column of water inside the plant builds a continuous column, which gives it a bit of strength. I mean, it's not nearly as strong as a plant with secondary thickening, but it still gets the job done, and it can hold mild plants up without making them fall. Another thing you might want to remember is that there are 3 methods how water travels inside the cells. There are 3 pathways, to be specific. One is called the apoplast pathway. The apoplast pathway is a system of interconnected cell walls. That means it generally moves water through the cell walls, and it can also send dissolved minerals like salts. And the other type of pathway is called the symplast pathway. Now, the symplast pathway goes through plasmodesmata to plasmodesmata. It doesn't go through the cell walls. Now, if you don't remember what a plasmodesmata is, if you don't remember the definition, it is the protoplasmic strands that connect the 2 adjoining cells. And that is called the symplast pathway. And finally, there is a vacuolar pathway. The vacuolar pathway goes through everything. That is how you need to remember it, and if you got that down, that is how water and material are transferred through the cell. So that is it for the plant cell, except that there are little structures that we didn't get into. So that is it for the cell wall. Now I'm going to go ahead and draw the other structures. So as you can see, we have a cell wall here with pits and plasmodesmata, and this whole thing inside of the cell wall is called the cytoplasm. So we're not going to draw anything specific to that. We're just going to go ahead and name it cytoplasm, and we're going to draw a large vacuole inside it. Now this vacuole is covered by a membrane called the tonoplast, and inside the vacuole there's something called the vacuolar sap. Now the adjoining part, now the two of them combined, the vacuolar sap and the tonoplast, is called the vacuole. And then we're going to go ahead and draw a mitochondria. The mitochondria is the powerhouse of the cell. That is how the cells generate energy. Then we're going to go ahead and draw the chloroplast. The chloroplast is usually the main structure which helps in photosynthesis. We're going to go ahead and draw some ADS ribosomes, and then we're going to go draw a Golgi body. The Golgi body secretes lysosomes, and don't worry, we're going to go into major detail later. We're going to go into the details of each and every one of these. And then we're going to go ahead and draw the endoplasmic reticulum, and finally the nucleus. The nucleus is where things get a bit complicated. We have the chromatin, the nucleus, the nuclear pores, the nuclear membrane, and the rough endoplasmic reticulum, which means that there are ribosomes attached to it. And then this whole structure is called the nucleus, with the nuclear envelope, the chromatins, and the nuclear pores, and the rough endoplasmic reticulum, and everything. And that is how you draw a plant structure. Now, before we go into definition of each of the parts, I'm going to go ahead and draw the animal cell as well. The reason that I'm going to draw the animal cell is because it also has some of the parts which actually are the same thing as the plants. It's not completely different. There are only very slight variations. The first difference you're going to notice is that it does not have a cell wall. Just like the plant cell, it has mitochondria, ribosomes, Golgi bodies, and it has things called peroxisomes. Now, in the plant cell, there are small structures just like that, but they're glyoxysomes, they're not peroxisomes. You need to remember that. There are microfilaments and microtubules, centrioles, which are arranged in a 9+2 structure. Don't worry too much about that, it's not really that necessary. And then there's smooth endoplasmic reticulum. The difference between the smooth endoplasmic reticulum and the rough endoplasmic reticulum, just a little note, I'm going to refer to them as SER and RER. The only difference is that the RER has small ribosomes attached to it. Then you can see a phagocytic vesicle over here, and then we're going to go ahead and draw the nucleus. The nucleus is just like that. It's full of chromatins, the nuclear envelope, the nucleopores, and then the RER. And this thing is called the centromere. And the entire covering of the cell is called the plasma membrane. Okay, so we have the structure of the entire plant and animal cell. Now what we're going to go into is all the little microstructures of it. The organelles, how they work and how they function. We're going to start off with something fairly common, is the plasma membrane. Now this is the structure that covers the entire animal cell, and it covers various membranes, and it is found everywhere. The plasma membrane is the outermost limit of the protoplasm. You can call it cytoplasm if you want to, don't worry about that. It consists of proteins, lipids, and carbohydrates. Now, most of the lipids in the plasma membrane are known as phospholipids. There's a little thing about phospholipids that you need to know. The phospholipids here contain 2 hydrophobic tails and a hydrophilic head. Hydrophobic means it tries to stay away from water, and hydrophilic is the opposite of hydrophobic. The plasma membrane was found by 2 people called J. Singer and G. A. Nicholson. Proposed a model structure for the plasma membrane. It's called the fluid mosaic model. It was called a fluid mosaic model because it has the ability to change and move around, and it was extremely flexible. We're going to go ahead and draw the structure now. It's very, very simple. First, we're going to go ahead and start with the phospholipids. We said that it has 1 hydrophilic head and 2 hydrophobic tails, and it's built by a complete long chain of phospholipids. But the thing is, it's made in a bilayer. That means there are 2 layers of phospholipids. And this time the head will be opposite because the tails are hydrophilic. So they try to stay away from contact of water, and the heads try to get in contact of water. So the heads are facing outwards and all the tails are facing inwards. Now, between these phospholipids, there are globular proteins, there are cholesterol molecules, there are intrinsic proteins, and there are peripheral proteins, and there are so many other things. There are glycolipids, there are glycoproteins, and there are large intrinsic proteins which have pores which allow molecules to pass through. This is the general structure, the fluid mosaic model of the plasma membrane. You have to know what the parts of the plasma membrane are. You need to know that J. Singer and G.A. Nicholson were the ones to find the plasma membrane, or the ones to term the plasma membrane the fluid mosaic model. You need to remember that it's termed the fluid mosaic model, and you need to remember what all the parts of the plasma membrane are. So now you know what it looks like. Just go through it again. If you guys have a hard time remembering what a structure looks like, the best thing to do is to draw it again and again until you remember it. I suggest that you guys should note this down. So the plasma membrane is about 0.1 micrometers in thickness. So we know what the plasma membrane looks like, but what is it there for? What does it really do? There are several functions of the plasma membrane. There are a few functions. We're going to go ahead and throw the definition out there because you need to remember the definition. I'm going to break the definition into 4 parts. You need to remember all 4 of these. It is a dynamic boundary of cells which permits the entry of water and ions and certain organic molecules. Now, the way it's cut down into 4 is that dynamic boundary is 1 mark, the permitting the entry of water is another, ions is another, and organic molecules is another. In the plasma membrane, there are 2 methods of transport. One is called the passive transport and the other is called the active transport. Now, as soon as I say passive and active, you need to realize that active takes energy and passive happens naturally. The passive transport happens through a concentration gradient, which means that it moves from a higher concentration to a lower concentration, much like anything. For example, if you take a drop of water and you put it in a cup and you turn the cup upside down and let go, the water will try to smooth itself out. So the water molecules try to spread around and give an equal distribution to the entire landscape. And then the active transport happens against the concentration gradient, because you could have active transport on a concentration gradient, but that would not really have a purpose because it would happen anyway without energy. A special thing to note is that glucose, nucleic acids, and amino acids are not soluble in lipids and do not simply pass through the phospholipid bilayer of a plasma membrane. In facilitated diffusion, which means in passive movement along the concentration gradient, and therefore carrier proteins are involved. Carrier proteins are used in active transportation. I'm going to go ahead and draw it here. We're going to see how this works. A carrier protein generally looks like this. There is a small pore which can take molecules, and what happens is, using energy, it turns around and around, and it takes a molecule from one side and it drops it off in the other side. But this doesn't happen according to a concentration gradient. What this does is it tries to take everything and let it pass through, even though it's against the concentration gradient. So of course, this is how active transportation works, and that requires energy. So the substances that dissolve in lipid diffuse easily through the cell membrane down the concentration gradient. The active transportation is used when they are not substances that dissolve in lipids, like glucose, nucleic acids, and amino acids. But that's how things enter the cell. How do things leave the cell? Most of the cells can carry out a function known as cytosis. Cytosis is an active transport method, and it's divided into 2 stages. One is called endocytosis and the other one is called exocytosis. Endocytosis is the transport of materials into the cell, and exocytosis is the transport of waste materials out of the cell. So these 2 processes get divided again into pinocytosis and phagocytosis. So solids are brought in and out of the cell, and that's called phagocytosis. And when it happens to liquids, it's called pinocytosis. So all the time we were talking about the functions of the plasma membrane. If you remember, the first function was that it acts as a dynamic boundary which permits the entry of water and ions and certain organic molecules. The second function is that it facilitates passive transport and active transport. The third function is that it regulates the exit of waste material. The whole process of exocytosis counts as an example or a function of the plasma membrane. The other function of the plasma membrane is that it allows to maintain an osmotic balance within the cell. An osmotic balance is a balance of water level because it can send liquids and solids in and out, and it can control the level of water inside the cell. So that is a really important part you need to remember about the plasma membrane. The fifth function is that it receives information through receptors, transmits signals, coordinates activities between the cell. Now, that was actually kind of a definition. You need to remember the 5 parts of this. The first one is it receives information, transmits signals, and coordinates differences between cells. Now, I'm going to give an example of that because that was quite a bit confusing and you can't really understand that the way I said it. So let's take an example now. Blood carries all types of hormones. These hormones recognize that they're target sites because hormones act on, well, depending on the hormone, they act on different locations. So they recognize it by the shape of the hormone. Each hormone is specific and it acts according to the receptor. So just like that, the plasma membrane facilitates that to happen. So that's another function of the plasma membrane. And we're going to the sixth and final function is cell recognition. Glycoproteins and glycolipids are cell surface markers. They help in the recognition by the shape and the branching pattern. Carbohydrates are unique to a person and they help in the tissue recognition. So there you have it. That is all for the plasma membrane. Don't worry, the lesson is far from over. Okay, so just a little recap on the plasma membrane. The plasma membrane is the outermost limit of the protoplasm. It was found by J. Singer and G. A. Nicholson, who named it the fluid, who said that the structure of it was a fluid mosaic structure. It contains 2 hydrophobic tails and a hydrophilic head arranged in a phospholipid bilayer with intrinsic proteins, globular proteins, cholesterols, and pores in the layer. And it has a lot of functions. Specifically, it is a dynamic boundary which permits the entry of water, ions, and organic molecules. It facilitates transportation through passive transport and through active transport. Glucose, nucleic acids, and amino acids do not dissolve in lipids, therefore active transport is used for that. Cytosis takes place in the plasma membrane as endocytosis and exocytosis. When it happens with solids, it's called phagocytosis. When it happens with liquids, it's called pinocytosis. The plasma membrane helps in maintaining the osmotic balance of the cell. It receives information through receptors, transmits signals, and coordinates activities between cells, and it involves in cell recognition. So that is it. That's all you need to understand about the plasma membrane, and then we're going to move on to the nucleus. So I'm going to go ahead and start by drawing a structure which is a bit more advanced than we started off. The nucleus is the largest organelle in the eukaryotic cell. It is covered by a double membrane. Remember this is a double membrane structure. It is not a single membraned organelle, it is a double membraned organelle. Usually cells have a single nucleus, but sometimes when it's like fungi, they could be multinucleated. The outermost membrane is continuous with the endoplasmic reticulum and often have ribosomes on its surface. That means that those endoplasmic reticulums are called rough endoplasmic reticulums. Remember that they don't have to have ribosomes. It could be RER or it could be SER, but generally it's RER. The nucleus is covered by the nuclear envelope. It possesses a large number of pores which we call the nuclear pores. These pores permit the passage of RNA and ribosomes that are formed in the nucleus. There is this jelly-like substance which is filled in the nucleus and it's called the nucleus sap. It contains nucleotides and chromatin. Chromatin is the material which is made up of DNA and histone proteins. It's when a strand of DNA gets tangled and tangled and tangled and tangled. the chromatins condense to become chromosomes during cell division. You might want to remember that because chromatins and chromatid and chromosomes, they get really confusing, the terminology. Don't worry, during that part I will explain what everything is. So chromatins are these long elongated structures that you can see, which are basically really long strands of DNA. So again, the chromatin is the strands of DNA, and when they condense and organize, it's called a chromosome. Inside the nucleus, there is one more thing. It's a very dark colored body, which is called the nucleoli or the nucleolus. It is bound by a membrane, and ribosomes are synthesized in the nucleolus. Ribosomes come out of the cytoplasm through the nucleopore, and the nucleus also synthesizes RNA. So I hope you got those stuff down. The nucleus is a bit complicated. It's the brain of the cell, double bound. It has a single nucleus, unless it's, of course, something like fungi. There are nucleopores. It possesses RNA and ribosomes. There's a nucleus sap inside the nuclear envelope. It has nucleotides and chromatins, which contain DNA and histone proteins. And there is a darkly colored body inside called the nucleoli. It is bounded by a membrane. The ribosomes are synthesized in the nucleolus, and ribosomes come out through the nuclear envelope pores, also known as the nuclear pores, and the nucleus also synthesizes RNA. So what are the functions of the nucleus? It has 4 specific functions. The first one is it stores the genetic material, that is by creating them into a code through DNA. It controls the cellular activities. Again, I did say that the nucleus is the brain of the cell. It synthesizes DNA to produce nuclei for cell division, synthesizes ribosomes and RNA required for protein synthesis. So if you had to write them down in short, this is what they would be. They store genetic material, it controls cellular activities, it synthesizes DNA during cell division, and synthesizes ribosomes and RNA during protein synthesis. Okay, so that is it for the nucleus. Now we're moving on to the mitochondria, the powerhouse of the cell. This is going to get really interesting because people believe that, well, when I say people, I do mean respected scientists, it could actually be a prokaryotic cell living inside the eukaryote. After we get into the details, you'll realize because it is a structure which contains a circular DNA and 70S ribosomes. It does fit the prokaryotic description. People do believe that it is a version it's in a previous evolutionary state to a prokaryote where it can't function as its own cell, but it could evolve into a cell of its own. So the mitochondria are organelles which are found within the eukaryotic cell. Just make that sure. If anyone asks, don't say that it is a prokaryote, because that's just speculation. It isn't proven fact. What we know is that it is a organelle of a eukaryotic cell, which is a double membrane organelle. It is around 1 to 3 micrometers in length, and it is how we get energy. When we learn about aerobic respiration, we'll realize how the mitochondria plays this massive role and how it provides ATP. But for now, we'll just stick with the structure. It is a double bounded membrane. It has these protrusions called cristae. And right now I'm going to go ahead and draw some circular DNA. And between these two layers, it's a fluid-filled space. I need you guys to remember that. And on the cristae, there are these little minute particles that are called stalked particles. And this entire fluid inside the mitochondria is called the matrix. It's not called the cytoplasm of the mitochondria. It's called the matrix. The terminology is something you have to remember. In fact, everything I said in this entire episode is, you need to remember that, because they actually give a lot of time. Even though we're going to do this in an hour or so, during school they give you like several weeks to study this, so don't worry about that. You can watch this video 20 times if you have to understand it, and go to LankanTutor.com and it will have the notes. If you want to go directly to the notes, it will be in the description below, so why don't you go ahead and click that and study. Again, the matrix carries the enzymes for the reactions during aerobic respiration, and it also contains the circular DNA and ribosomes. Now, we do know that ribosomes are present in prokaryotes and eukaryotes. In prokaryotes, they are 70S ribosomes. In eukaryotes, they are 80S ribosomes. But inside the mitochondria, which is inside the eukaryote, they also have 70S ribosomes. It's the same ribosomes that you can see in the prokaryote. So these ribosomes are used to synthesize proteins. The matrix carries enzymes for the reaction of the Krebs cycle. Again, Krebs cycle is a part of the aerobic respiration. It's called glycolysis, Krebs cycle, and the electron transport chain. Don't worry about that. During those three parts, the Krebs cycle is the part that happens in the mitochondria, and it helps create the ATP. It doesn't necessarily create the ATP. It creates around 2 ATP molecules. Most of the ATP is created in the electron transport chain, but that can happen because of the Krebs cycle. Don't worry about that. There is this entire part about cellular respiration, so we'll learn about that later. So that is it about mitochondria. The only thing you need to know now is how to be able to draw the mitochondria and how to be able to mark the parts. You learn a lot more about the functions of the Krebs cycle and the substrate phosphorylation and everything later when we're talking about that. It's at the very last part of the second unit. And then we go on to a rather peculiar structure called the Golgi complex. There are these long tube-like structures in the eukaryotic cell, and they're close to the nucleus in the cytoplasm. If you want to define its shape, they are stacks of flattened vesicles. Again, I forgot to tell you guys the shape of the mitochondria. If they actually ask you to tell the shape, they expect you to write it is a sausage-shaped organelle. That's actually what they expect you to write. So the Golgi body, the Golgi complex, let me just say that, the Golgi complex, you can call it Golgi bodies, you can call it Golgi complex, you can call it Golgi complex, it really doesn't matter, as long as you can write it for the paper. So if you're thinking that the Golgi complex looks a lot like the smooth endoplasmic reticulum, it's because it's made from the SER, or rather it's synthesized from the SER. And likewise the Golgi bodies and likewise the Golgi vesicles secrete these phagocytotic vesicles, which are called lysosomes, which is the next organelle that we're going to be talking about. But before we go there, let's talk a bit about the functions of the Golgi bodies. Proteins and lipids manufactured in the ER, or the endoplasmic reticulum, are transported to channels of ER or vesicles into Golgi complexes. So this lipid and proteins combine with polysaccharides to form glycolipids and glycoproteins. So you need to know that the Golgi also aids in the formation of glycolipids and glycoproteins because of its ability to transport. When necessary, the vesicles containing glycolipids and glycoproteins are budded off, and these structures are used in the distribution. The Golgi complex helps in the phagocytosis of the cell. So that was a little complicated. Let's just run it down into 4 little points that you need to remember. Just memorize all 4 of these and you should be good to go. So the first function is the collection of molecules synthesized in the cell, the packing of molecules synthesized in the cell, the synthesis of glycoproteins and glycolipids, the distribution of molecules synthesized in the cell, and the formation of lysosomes. Even though that seemed like a lot, if you take out the formation of lysosomes, it's generally the collection and packing and transport functions of the cell. And don't forget that it synthesizes glycoproteins and glycolipids. And with that, we are done with the Golgi complex. We can move on to the next part, which is actually the endoplasmic reticulum. Now, the endoplasmic reticulum is a network of internal membranes from flattened tubular sacs. It separates the cytosol, the cytoplasm, from the ER lumen continuous with the outer membrane of the nuclear envelope. So you got that, right? It is a network of flattened tubular sacs which cover the nucleus. That's generally what they're trying to say. If you remember that we said that there are two types of endoplasmic reticulums. One is called RER, meaning rough ER, and the other one is called SER. The only difference between the RER and the SER is that the RER contains ribosomes. That is the only difference, and if you have a problem remembering that, just remember it as RER. Well, R means ribosome. Just think of it like that. That would be easy to remember, but it's called RER because it's called rough ER. Now we're going to divide it into two and study about each of them individually because the functions they have is different. The RER or the rough ER are still flattened sacs and ribosomes are bound to it. It synthesizes membrane phospholipids and membrane glycolipids. They transport enzymes and other proteins within the cell and produce transmission vesicles. That means lysosomes. Then we go on to the smooth endoplasmic reticulum. It's a network of tubular sacs without ribosomes attached to it. It synthesizes lipids, steroids, and carbohydrates. Also, these molecules are transported within the cell through transmission vesicles. Examples, the male hormone testosterone and steroids, but the SER has some special functions too. It causes detoxification of toxic substances, and the SER stores calcium ions. You want to remember that. And finally, the most important part of the SER is that the SER is what makes Golgi bodies. Remember that the RER does not make Golgi complexes, it's the SER. So we're going to talk about the next organelle. It is the lysosome. The lysosome is a phagocytotic vesicle. Lysosomes are created by rough endoplasmic reticulums and Golgi complexes. They are the organelles which destroys dead matter, and they help in the phagocytosis of the cell. Even though I explain it again and again, you're going to have to study the definition, so I'm going to go ahead and say it. They are produced by the Golgi. They are a membrane-bounded vesicle contributing to the digestive activity. They are spherical in shape, contain hydrolytic enzymes which catalyze the breakdown of carbohydrates, proteins, lipids, nucleic acids. So what they're trying to say is that they have enzymes which break down the molecules like carbohydrates, proteins, and lipids. It generally destroys unwanted material and it breaks down molecules into smaller forms. It catalyzes things. So exocytosis, endocytosis, all of them happen through lysosomes. I'm going to go ahead and give a little diagram of how endocytosis takes place. Now, when a material enters the cell, the lysosomes have the ability to act on it. It has these enzymes which break the walls of the cell, and it completely digests and absorbs the matter, and then the residual matter, or the stuff that's left, the unwanted parts, is completely removed through exocytosis. So that's how the lysosomes help phagocytosis. The main function of the lysosome is cellular digestion. It digests food particles received by phagocytosis, as we discussed earlier. They digest worn-out organelles such as other mitochondria, other endoplasmic reticulum, Golgi bodies. They make way for new ones while recycling material locked in the old organelle. So the lysosome is kind of like the recycler. It takes all the garbage and pushes it out of the cell. It also transports leftover matter through exocytosis, and it has a function called autolysis. This is the way that the lysosomes start digesting material and breaking them down. It's what causes cell death. In frogs, when they're at the early stage before metamorphosis, they are tadpoles and they have a tail. And when they grow up, their tail falls off. Now, for the tail to fall off, now it is a complete part of the it is a complete part of the body of the tadpole. So what happens is those hydrolytic enzymes, they start digesting the tail and they completely break it off, and it causes the cells between the tail and the body to die off, and it completely falls off. That is what happens during autolysis. And another example is there is a specialized lysosome called the acrosome. The acrosome is found in the head of the human sperm. Because of that, it has the ability to penetrate the egg of the female. The shell is dissolved and the sperm gets to go inside the egg in order to fertilize it. That's all you need to really know about. Try your best to memorize all of that. And then we go on to microbodies. Microbodies are not, you don't really need to know so much on microbodies. Just remember that there are two types, peroxisomes and glyoxysomes. Glyoxysomes are present only in plant cells. Peroxisomes are present in plant and animal cells. These microbodies are produced by the endoplasmic reticulum and are membrane-bound vesicles, and they contain oxidizing enzymes. They are spherical in shape and they are smaller than lysosomes. So if you had to mark a structure and you see this big circle, that would be the lysosome, and if it is a smaller circle around it, that is probably the microbodies, the peroxisomes and the glyoxysomes. The enzymes in the glyoxysomes convert fats into carbohydrates. The enzymes in the peroxisomes catalyze the breakdown of hydrogen peroxide. That's pretty easy to know. Peroxisome, hydrogen peroxide. Glyoxysome, fats into carbohydrates. And also, detoxification of peroxides occur due to peroxisomes. That's all the things you really need to know about that. I'm not going to go into detail, generally because you'll probably be wasting your time. If you would like to study about a bit more about the microbodies, I'll be sure to add a bit more in the written note. You may be thinking that this section never ends because it is a rather long part. I did tell you this will probably be the longest video in this entire syllabus. Next we move on to the chloroplast. You've heard the word chloroplast before and you know what chloro means. Chloro means green. So by now you should figure out what chloroplasts are, and it is a double membraned organelle, just like the mitochondria, and people believe that could be a prokaryote as well. It has 70S ribosomes, but it is a bit specialized. It is only found in plant cells. It has circular DNA. It has these weird flattened sacs which are called thylakoid, and they are connected to other thylakoids by a structure called grana. They have ribosomes and a stroma and a starch grain. Integral lamella, it has the outer membrane, the inner membrane, and it has lipid droplets, and also a lot of ribosomes in one place. It's called a polysome. So let's go through that again. The chloroplast is a cell organelle in plants. It is a double membrane structure. It has an internal membrane in thylakoids. These are flattened sacs filled with fluids. The thylakoids are stacked to form grana. The surrounding areas of the thylakoid is called the stroma. Just like the matrix of the mitochondria, you call it the stroma of the chloroplast. It's just a matter of terminology here. In addition, the chloroplast has starch grains, lipid droplets, ribosomes, RNA, circular DNA. And in the granum of the thylakoid, it contains photosynthetic pigments called chlorophyll A and chlorophyll B and carotenoids and so on and so forth. Now remember that photosynthesis happens because of the chloroplast. You'll learn a lot more about photosynthesis in the latter parts of this unit, but for now just remember the structure of the chloroplast and try and make sure that you can draw it. The reason that I really didn't go into massive detail on the mitochondria and the chloroplast is because of the fact that you're going to have to study them again during photosynthesis and cellular respiration. But I will give you the basic structure and how it will work. So other than what we've discussed now, the main function of the chloroplast is photosynthesis. That is how light energy is converted into chemical energy in the presence of carbohydrates. Chloroplasts are found mainly in the palisades and the leaf mesophyll. Another thing is that don't quote the definition of photosynthesis here, because that's nowhere near what you need to know. You need to know 5 really long points of photosynthesis. We'll get to that later. So that is it for the chloroplast. don't worry and think that we're skipping out on one of the most important organelles, because we're going to be getting back to that later. The next part is the cytoplasmic skeleton. So what exactly is this cytoplasm that we keep on talking about, and the cytoplasmic skeleton? In a few words, it is the internal framework of the cell. It's what gives the cell the shape, and it's what keeps it together. In all the eukaryotic cells, the cytoplasm contains an internal three-dimensional lattice, the cytoskeleton. It is the supporting structure of the cytoplasm, and it's made out of microtubules and proteinous filaments, and it is a dynamic structure, breaking and deforming as needed. And there are three types of components in the cytoplasmic skeleton. So remember it is like this meshy structure that gives shape. The actin filaments help in movement, the microtubules help in transport, and the intermediate filaments help in support. The actin filaments are composed of two proteinous chains which grind around one another to look like this. These are responsible for muscle contraction, and in plant cells, the movement of the cytoplasm and the movement of the white blood corpuscles happen because of actin. Don't worry, there is a part about how the muscle contraction works and the actin myosin sliding filament theory. We haven't gotten there yet, because in this part that we're talking about, we're only really focusing on the structure and function of organelles as well as an organelle, instead of how it functions as a complete system. The second type of thing that you can find in the cytoplasmic skeleton is the microtubules. Now, these are completely empty tubes composed of proteins, and they are circular. These are used in the intercellular transportation within the cell. So the Golgi vesicles are transported, and everything moves throughout the cell using the microtubules. All you need to really remember is that the actin filaments help in movement and the microtubules help in transport. And finally, we go into the intermediate filaments. They are proteins, and they are the most durable elements inside the cytoskeleton. It is made out of extremely tough fibrous proteins. The molecules are in a rope-like manner, and it gives tremendous strength. The proteins present in are keratin and vermetin. Those are extremely strong fibrous proteins, and they don't break. So here are the functions of the intermediate filaments. They give strength, support, and they help in the movement. And next we move on to the centrioles. So the next part we're going to be talking about is centrioles. The centrioles are a weird little organelle because they're actually made from the connection or the formation of a bunch of microtubules. If you don't know what a microtubule is, I have bad news for you, buddy, because we were actually talking about microtubules like 2 minutes ago. The thing about these microtubules is that they join, and 9 of them form to make a hollow circular ring, a hollow circular cylinder to be precise, and they are perpendicular to each other, and they're in triplets of 9. The cylindrical structures are perpendicular to each other and are only found in animal cells. Remember that plants don't have centrioles. So the main function of the centriole is that it helps the organization of the microtubules and in the aster and spindle formation of cell division. You won't really need to know anything other than that. In the centrioles, it's a rather small amount that you need to know. Just remember that it helps the aster and spindle formation during cell division. And if you're like, well, I don't know anything about cell division, don't worry, cell division is coming up soon. Well, when I say soon, I mean in the middle of the entire second unit, so that would be a long time from now, but it's coming up. Next we have cilia and flagella. The cilia is found in free surfaces. Cilia is generally like hairs, and flagella is a locomotory structure that we talked about before. For example, we said that in prokaryotes, some of them have flagella which help them move. But how is flagella formed? It's also formed from microtubules, which are found in the cytoplasmic skeleton. But the difference here is that Unlike in the Centrioles, which had a arrangement of triplets of 9, this is a 9 plus 2 arrangement, which means that they are 9 duplets. The main function of flagella is for locomotion and the transport of material in the cell surface. So I'm going to go ahead and draw this, the cilia. How you know it's a 9 plus 2 arrangement is that it has 9 around it in the basal body, and there are 2 in the middle, and they're all in duplets. So we've come a very long way, and we're almost near the end. We have to know just the vacuoles, ribosomes, and the cell junctions are left. So let's start off with the vacuole. Is a large structure which is bound by the tonoplast. Like we discussed before, it is filled with a liquid which is known as the vacuolar sap, which is found in plant cells. In mature plant cells, the vacuolar sap occupies a huge space. It stores water and other materials like sugars, ions, and pigments. The function is to maintain the water balance by removing and accepting water into the cell. It gives turgidity and strength to the plants. It produces colors in some plants by having pigments inside the vacuolar sap, and it stores soluble substances that are needed for cellular activities like sugars and amino acids. That's really all you need to know about the vacuole. It's this large storage unit. And then we move on to the ribosomes. We were talking a lot about ribosomes, but we really didn't know what a ribosome was. A ribosome is generally like this: a large unit next to a smaller unit, and the ribosomes are made from rRNA. The raw material for ribosomes would be ribosomal RNA, like we talked before during nucleic acids. And also proteins are used in the formation of ribosomes. And remember, there are 2 types of ribosomes, ATS ribosomes and 70S ribosomes. The ATS ribosomes are found in eukaryotic cells. The 70S ribosomes are found inside prokaryotic cells and also in organelles like mitochondria and chloroplast. The functions of the ribosomes are the synthesis of proteins coded by mRNA with the help of tRNA. We actually did a lot about ribosomes, if you remember, during protein synthesis in the earlier parts of the second unit during the elemental composition. If you don't remember that, you might want to check that video out again. The thing is here is to watch it again and again and again until you make sure that you remember everything, because we need to remember everything if you are to have the slightest chance of getting through this exam properly. So we're at the final part, is the cell junctions. Now you need to remember that in plants, we said before that the 2 cytoplasms of the plants are connected through a plasmodesmata, a hole, if you will, between the 2 cell walls which exchange material. But how do animal cells join? Now there are 3 junctions. I'm going to go ahead and start drawing them now. There are 3 junctions in the animal cells which helps the connection of them. And they are called the tight junction, the anchor junction, and the gap junction. And they are found in the plasma membrane of the cell. I'm going to go ahead and just give the definitions of the 3 junctions and tell you the function so that we can get over with this. The tight junction connects the plasma membrane of the adjacent cell tightly. Protein materials are used to connect the plasma membrane. It is bound tightly around the cell. It forms a leak-proof fibrous protein belt around the cell. Remember that. It forms a leak-proof fibrous protein belt, and it is extremely tight. And for example, in epithelial cells, it prevents the leakage through the intercellular spaces. So the tight junctions are used for leak-proof situations. Then we go to the anchor junctions. It is a mechanical attachment of the cytoskeletons of 2 adjoining cells for strong binding. Remember that. For the example for that would be the skin epithelium. Here 2 proteins are used, and these junctions are found in locations which have mechanical stress. And finally, the gap junction, also known as the communication junction, it allows signals and materials to pass through one cell to the other through a direct connection, which means that there's basically a hole between the 2 cytoskeletons. It's kind of like the plasmodesmata for animal cells, if you will. For example, you can see them in excitable tissues like the heart muscles. In these, electrical signals pass through the junction. Now, the functions of these three junctions would be the exchange of materials between the adjacent cells, the prevention of fluid leaks between the cells, allowing the cells to communicate, and also allow tissues to work together. That is it, guys. We have actually finally completed the organelles. So we're going to go ahead into this one slight revision. Don't get mad. We're going to go ahead and compare the plant cell to the animal cell. The main differences is that the plant cell has a tough cell wall. It has pits and plasmadesmatas. It has a middle lamella which joins the walls of the adjacent cells. It has chloroplast, and in mature plant cells it has a single large vacuole filled with a vacuolar sap, and centrioles are not present. The storage material is starch, and cilia and flagella are absent, and the only types of tissues that could divide in the plant cells are called meristematic tissues. Now, the counterpart for that of animals would be it would not have a cell wall, there are no pits, and there are no plasmadesmatas, there is no middle lamella, plastids are absent, there is no middle lamella, and there is no chloroplast, there is no vacuole, but there are centrioles, and the storage material is glycogen, not starch. Cilia and flagella could be present in some forms. It's not always present, but it also could be present. And most cells, except the nerve cells, have the ability to divide. That's why you shouldn't kill your brain cells, because they can't recreate. So thank you guys for watching. It has been an extremely long process, and my throat is hurting pretty badly now, and we will leave it off for here, and we will be going next time into more detail of the plant tissue. So until next time, have a great day. If you want to study this, I know that there is no way you understood everything that I said today. It's because you can't. They actually allow several weeks at school for you to understand what we did in this past hour. It's insane. And I want you guys to go to LankanTutor.com, read the note around 5, 6, 20 times if you have to, until you go to a point that you can pretty much say everything without looking at any of the notes. If you can, congratulations, round of applause to you. And if you don't remember and you want to remember it again, and if you want to go through it again, go ahead and watch this video again. Watch it as many times as you need, because trust me, this is not easy shit. Until next time, have a great day.